Hi everyone, thanks for listening to my presentations. Today I'll be talking about thyroid hormone synthesis, and this will form part 2 of 7. If you haven't listened to part 1, which was all about thyroid gland, the scope of this presentation, and the brief anatomy of the thyroid gland, please do. And with that, let's go. Okay, the question we should ask ourselves is, why burden ourselves at all about thyroid gland? Well, I have the answers. We will bother ourselves because thyroid gland has hormones that are very useful to everybody. Like in children, it determines brain and somatic development. And it also regulates metabolic activities in adults. The hormones here will affect the functions of all organs. And initially, very large quantity is produced and stored, but will be released in the circulatory system as the metabolic activities or the functions of organs or the development of brain and somatic development in children will require. Biosynthesis and secretions. So synthesis and release or biosynthesis and secretions are maintained by a regulatory mechanism. And that regulatory mechanism is very sensitive to minute changes in circulating hormone concentrations. All we are targeting is euthyroid situation. Okay? Anatomically, thyroid gland has spherical follicles, each containing a single layer of follicular cells. And there is a lumen filled with colloids called Thyroglobulin. The Thyroglobulin is a glycoprotein produced by the follicular cells of the thyroid gland. The thyroglobulin form the main precursor to thyroid hormones. The thyroglobulin tyrosine, in addition with iodine, and will later cleave. Then the tyrosine iodination is catalyzed by thioperoxides in the follicular colloid. Thyroglobulin acts as a substrate for the synthesis of the thyroid hormone. T4 and T3 are tyrosine and triiodothyronine respectively. The inactive thyroid hormones and iodine are stored within the follicular lumen. T3 and T4 are attached to the thyroglobulin and form the colloid within the follicle. Thyroid-stimulating hormone leads to the cleavage of the colloid by proteases to release the thyroglobulin from its T3 and T4. T3 and T4 are the active forms released into the circulatory system. Thyroglobulin is then recycled back into the follicular lumen. For what? To be used again. Then the T3 and T4 can be unbound or bind to plasma proteins. T4 forms about 90% of the total thyroid hormones, and it has 2 iodine atoms on its phenyl or outer ring, while T3 has only 1 iodine atom. If an iodine atom is removed from T4, then T3 is formed. T3 is about 9% of the thyroid hormones and is 3,5,3 triiodothyronine. But that's what we call reverse T3. That will form about 0.9 to 1% of the thyroid hormones and it is reversed in the sense that instead of being T3 with 3,5,3 triiodothyronine, we are going to find 3,3,5, not 3,5,3. So it is reversed. But we don't need to bother ourselves about that too much because it is biologically inactive. Most of this reverse T3 is not from the thyroid gland. Well, with that, I've come to the end of this very presentation. The next is going to be about part 3 of 7, and that will be all about the role of iodine in thyroid hormone synthesis. I believe strongly that we'll be interested in knowing the role of iodine in the synthesis of these thyroid hormones. Thanks for listening. Kindly remember to share and subscribe. I appreciate it.